All right, so welcome back. Here we go again. Back by popular demand, right? Okay, so this is going to be the start of the Glycocalyx ESL sort of discussion, right? So this is going to have to evolve a bit as we sort of go through. But what I thought we might do is just do a really quick recap. We've done the discussion around nanobubbles and the importance of nanobubbles, and so anyone watching this who hasn't seen that video should definitely watch that video first, because we're going to refer to a lot of the concepts and implications we spoke about there. But just from the point of view of recap, we probably actually just want to flick through a couple of basic bits of nanobubbles. So the basic premise, I guess, is that nanobubbles form, they're present throughout the body. And the reason we believe that they're stable is because of the effect of the bubble-bubble inhibition, the coalescence inhibition. And that's as a result of either different salt concentrations. In the case of blood, it's sodium chloride salt concentrations. And this, we know it around that sort of 0.175 molar, we get this sort of change in coalescence inhibition. We also see a similar thing with certain sugars, okay? Again, not all sugars do it, some do, some don't, similar to salt, and also through different amino acids. And this one we're looking at tyrosine, which forms very fine bubbles in particular. So nanobubbles are an important part of the message. And one of the other key things, which I think we might sort of start off with, is just a recap around the importance of dissolved gas and what that means from a physical chemistry perspective. We spoke about the fact that if we remove dissolved gas, emulsions become stable, hydrophobic interactions disappear, and those enzyme substrate reactions we spoke about, they all sort of stop. And even the measurement of things like pH, buffers and molecular forces, they all change. And the really good macro version, I suppose, of that is cavitation. And we'll go through that cavitation video, I think, really to sort of show that. You spoke about the fact that if we degas water, cavitation stops, because cavitation is the forming of micro and nano bubbles, which ultimately corrode or destroy the propellers in cavitation. Is that a fair sort of summary?
Yeah, I guess so. It's much more subtle than that. The examples you talked about are very graphic and very obvious. Like the cavitation of a propeller makes a lot of noise with the outboard motor. It's a macroscopic manifestation of these tiny nanobubbles that occur naturally. And the bubble fusion problem, if you look at the ocean, it's foaming. Bubbles don't fuse. And if you look at freshwater foam, the foam doesn't exist, the bubbles do fuse. And that depends on the salt and the type of salt and the type of solute. Well, we've inferred from that over the years that nanobubbles are the source of those kinds of effects, things like hydrophobic interactions and so on, enzyme interactions. But now there's a lot more direct evidence that they do exist, and they depend in very subtle ways on salt, on amino acids, on sugars and so on. And they're the fourth element. We look at theories that involve water, matter, salt, oil and so on. And we've ignored the dissolved gas, which turns out to be crucial. And it turns out it's got the capacity to form all kinds of structures, one of which is nanobubbles. And when they form, they're energized and they drive the chemical reactions. So there's direct evidence for them now. They're not just made up, inferred.
They're not just speculation.
They're not just speculation. They're actually observed with laser scattering, with conductivity, with all kinds of ways.
So as the measurement techniques have become more refined and we're able to see smaller, if you like, for want of a better term, then is that what you're referring to?
Yes, and also people have missed them because they didn't want to see them. There was no reason to think before that gas was organised and had a role to play. People didn't think it was possible because the amount of dissolved gas from the atmosphere in a liquid, in an electrolyte salt solution, is very small. So you think, well, that's irrelevant.
And it's sort of, we spoke about it's also a constant in the sense that unless you're specifically degassing solutions, then there's going to be gas in those solutions.
Yeah, it's constant in the biological system. It only shows up if the salt concentration changes and you get sick.
Yes.
And that's when it shows up.
Yeah. Okay, so I just want to touch on one of those points that you spoke about, the fact that they sort of, dissolved gas has a role to play, and there's lots of different ways that bubble, coalescence inhibition may be altered, if you like, with sugars and salts and those sorts of things. But I guess one of the other really important things is the type of gas also, and probably the best example of this we've seen is carbon dioxide. So it's not just the fact that if there is dissolved gas, it's got a role to play, but even different gases play a different role, or can play a different role or have different effects. And we've definitely seen that with CO2 from a sterilisation point of view. So maybe if we start with that and the Mark Adams discovery in particular, and the work that Adrian's done around CO2 as a specific inactivator of viruses, etc.
Yeah, well, how to explain that? The nanobubbles are not different in terms of the way they affect macroscopic phenomena like macroscopic bubbles, but the gas themselves, carbon dioxide is one that's very subtle, and it's known to be very subtle in physical chemistry all the time. Carbon dioxide in the atmosphere is very low concentration, but it changes the pH of water from 7 to 5. It makes it acid. Mm-hmm. But what's going on really is that inside that CO2 gas bubble, the way it's interacting with the surface is quite different to the way that such a gas bubble with oxygen or nitrogen interacts. It's mainly the interaction with the surface that causes the reaction, which is very different from one gas bubble to another. Mm-hmm. Like carbon dioxide, we know macroscopic bubbles of that affect pathogens like bacteria and viruses. And the first instance of the first good experiment on that was done by a guy called Mark Adams in 1945 or 1948.
48, I think, yep.
48. Yep. And he just shook up a suspension in physiological medium of bacteria and viruses and found that the mere shaking of it killed the viruses. And he attributed that to the exposure of the coats of a virus, which is made up of a close-packed bunch of proteins, to the air, which destroys the coat. And in this case, was due to carbon dioxide. We, much later, did experiments recently where we're trying to sterilise water, which has pathogens, mm-hmm. like E. coli, and we're using a bubble column that's just using gentle bubbles that pass through a frit. The bubbles come up, they don't fuse above physiological salt concentration, the macroscopic bubbles, but what keeps them from fusing are a bunch of nanobubbles. So the nanobubbles are what's reactive. So those nanobubbles that occur on the interaction with the macroscopic bubbles, imagine a fish tank with bubbles going up, as they come near the surface of a pathogen, the nanobubbles interact with it and they react with the thing and they destroy the protein. That interaction with the protein, which is it's a bunch of folded polymers, that interaction with the gas bubble causes it to unfold. It's a hydrophobic interaction. It unfolds it, and so it's destroyed. It doesn't have the
It breaks the code of the virus.
code of the virus, and so then it exposes more of them. Yep. So the coating's lost, and the RNA of the virus is destroyed. So that's what we think is going on.
And sorry, and just to be clear, then, so what it sounds like you're saying is it's not the micro bubbles themselves, but the nano bubbles. You know, I'm sort of seeing in my own mind a sort of a whole bunch of nano bubbles. So now we're talking about sort of nanometer to sort of, you know, 10, 20 nanometer, that sort of size. Yeah, that's the sort of size it went. Yep.
2 to 3 nanometers.
Yep. So very, very small bubbles. Those tiny bubbles in between what might be 50 to 100 or larger nanometer micro bubbles. Yeah. And it's those nano bubbles, the interaction of those, that's causing these reactions, or causing these proteins to essentially unfold or separate and expose the RNA from inside the virus. Yeah. Okay, and we know this in the sense of the work, particularly that Adrian did with the piggery waste, etc. We know that CO2 has a very different effect to nitrogen, to oxygen, etc. And there's therefore a very specific, gas-specific effect of that, not just the exposure to thermal energy.
Yeah. The other bubbles have different kinds of effects, but we can come to them later.
Yeah. Okay. So, all right. So, let's at least kick off the sort of the main sort of point of all this, and this is our sort of Glycocalyx endothelial surface layer, and this is really, I guess, the culmination of a process you went through in trying to understand from a self-assembly perspective, and this sort of molecular forces angle is the way I kind of look at it, is trying to understand this biological structure in the body. So I think let's first of all talk about exactly what we mean by the glycocalyx and the endothelial surface layer, because like many things in biology and medicine, they don't seem to be, well, at least consistently defined, right? So we've, you know, as we've found in going through the research, there's lots of variabilities of endothelium versus endothelial cells versus glycocalyx, etc. So I want to start with getting specific of what we actually mean, what we're referring to. And this is one of our favourite pictures that from Hans Vink and others, that is really a sort of, you know, the best sort of close-up. So this one's actually looking at a rat capillary. And so in here we've got this sort of a number of different sort of bits to this. So this sort of dark ring here, or this section down here, is essentially a mess of polymers, and we'll come to that in a second. That is sort of like coats the cell walls, if you like, the sort of 3 nanometer bilayer of the cell wall. So we've got this glycocalyx sort of structure sitting on top of there. And then some of those strands, for want of a better term, seem to then sort of poke up into in this case, into the blood vessel itself, and form this kind of like, you know, gel-like structure is the way it's often referred to, with these sort of strands. And this bit here is, I guess, what we're referring to when we talk about the endothelial surface layer, is this layer that includes these strands and the sort of gap above those. And the glycocalyx is actually more this kind of bottom sort of section, which is, you know, where the strands are sort of running more sort of horizontal, for want of a better term. But is that a fair way to describe what we're referring to?
Yes, the endothelial surface layer has been known for a long time, very long time, for 150 years. It's a fuzzy layer. I just call it a fuzzy layer. And nobody knew what the structure was. When they tried to look at what was in this layer, which is very thick, very wide in extent, it's a micron in thickness, up to a micron. A micron, that's 10,000 angstroms. That's a very long way. It's So
again, just to go back to, we used that analogy last time we talked about I talked about the rugby field. That's the way I like to look at it. You've got this 100 meters of field. You've got this sort of up to say 10 meters of sort of in goal area, and then you've got this dead ball line, this white line at the back of the very back of the field, you know, and conceptually that's your cell bilayer, that's your lipid bilayer of your cell membrane, is that just that single white line at the back. But even to get to that, you've got the in goal area, which is this sort of glycocalyx sort of mesh of polymers, which we'll get to in a minute. But then you've got this endothelial surface layer, which is the rest of the field, the 100 metres you've got to get to first.
Yeah, no, when they tried to measure what was in it, they could find nothing in it.
Mm-hmm.
So it's very dilute set of polymers sticking out like a bunch of seaweed from the surface. And it flows with the seaweed too. It's not really a gel, it's very loose, and it can flow with the flow of blood backwards and forwards, and the blood flows through it. But it's drawn out, it's teased out of what's called the glycocalyx. Again, that's been known for a long time. And the glycocalyx is really solid polymers that are structured, and they're porous, like a Swiss cheese, connected tubes. Mm-hmm. And that's also very thick compared with the bilayer on which everybody focuses. The cell membrane, a bilayer of lipids, 2 layers of lipid molecules, like in extent like an olive oil molecule, 2 layers of olive oil per molecule. That's only 3 nanometers, 30 angstroms. It's 10 layers of hydrogen molecules. It's very tiny, but it's invested with all the duties of the cell membrane. And for nervous cells, it opens and closes channels and lets sodium and potassium in. So it's invested with all these very complicated duties, whereas the glycocalyx, which is a much thicker and stronger thing, had no structure at all. Nobody knew. It had to do something, but it did nothing specifically. So this whole structure, the endothelial surface layer, which they couldn't see until recently, and the glycocalyx, are very obviously very important, but nobody knew anything about them.
And just on that, here's another. So this is an example of this is a bacteria, an image of a bacteria, and again, you know, we can see in this bacteria. So you've got this sort of like, here's your cell membrane on the outside, and then you've got this sort of thick, fuzzy, you know, thick layer here within this sort of fuzzy layer sort of sitting on the outside. So you've got this entire cell. And again, but I guess one of the problems with this is because we sort of look at things largely in 2 dimensions, we often lose sight of the fact that what we're looking here is a 3-dimensional thing, not a 2-dimensional thing. But we can see that the entire outer layer is covered by this, you know, in reality, compared to this tiny cell membrane, you've got this enormous structure sitting on the outside of these cells. So that's what we're talking about specifically. And again, if we sort of okay, so
bacteria can be more complicated membrane.
Sure. And I guess, and just to be sort of clear, I guess when we're talking about the ESL and GC, we're talking about a type of structures as opposed to necessarily a very specific thing. The GC-ESLs varies to some degree between different cells in terms of thickness and some of those sorts of things, but almost every cell has this fuzzy layer or has this sort of, you know, outside coating that, and the connectivity of that. So, so this is our sort of like, you know, cartoon image sort of diagram. So we might sort of go through this in a bit of detail to give a more specific idea of what we're talking about. But I guess just to be clear, what essentially I think the message that you're trying to get across is that this glycocalyx, the way I like to look at it from what you've explained, is it's a series. First of all, it's based on a dynamic self-assembly process. So here we've got different types of, you know, lipids and proteins, and we've got water, we've got salt, we've got a whole interaction of lots of different components that, because of those interactions, we'll get into why you think this as we go through. But what we end up forming here is a set of very dynamic self-assembled tubes, for want of a better term, that are very substantially involved in cell mediation in terms of not just cell traffic, in and out of the cells, but also in and around the cells and the way the cells connect and communicate with each other. So you believe that they're actually an expansion of the neural network, because, and we'll get into to why, but there's a electrical sensitivity of the glycocalyx that's part of the story itself. But the ESL GC has a number of very important roles. One is to act as an exclusion zone and to sort of help protect the cell, and we'll give some examples of that, from pathogens and things that we don't want sort of penetrating or disrupting that cell membrane. But also much of the activity that's, I think you're just alluding to, that is assumed to happen via or through the cell membrane, in fact, is more likely to be substantially, if not totally, a function of the glycocalyx itself. So,
well, it has to be, because it's very thick, and compared with the cell membrane, that figure's a little bit misleading, because you can see the lipid bilayer there on the right. It looks big, but it's only 3 nanometers.
We have to make it viewable.
Yeah. But and similarly, the glycocalyx, the pink area on top of the yellow line,
to make it fit
is much thicker.
Yeah. So it's a logarithmic scale, shall we say?
Not
even to scale. So so here we're talking about something sort of 3 nanometers. This is sort of 17 times that.
But the structure is made up of sulfated polymers. There's 4 main ones, and they've got a hydrocarbon backbone on which there are sulfate molecules, or sulfated molecules, and they're familiar words: heparin sulfate. You can buy them at the chemist. Chondroitin sulfate, and there's several others as well. But the sulfate molecule is very special. It's charged. It's got a divalent charge, and because of the charge, it's very hydrophilic. It likes to bind water. So you've got a hydrocarbon region of the polymer, which is oil-like, and you've got a hydrophilic water-like area of the sulfate. So it's in a situation where it wants to minimise the contact of the sulfate with water or with the hydrophobic region. So it forms a coil. So it coils up. So it forms a helix.
It's a helix, yep.
It's a helix. Yep. So the helix protects the sulfate. It's pure water. It can pass electrolyte through it. The blood is passing through it, but not the contents of the blood, not the serum albumin and all the other contents of the blood, just the electrolyte.
Okay, so let me just sort of walk back, so I think we're clear in our mind what's going on. So these sort of like wispy lines we have here, what we're really alluding to here is, if we imagine a big long polymer with a whole bunch of sulfate head groups on there, what it sounds to me like what you're saying is, because of that affinity with water and the fact that we're in essentially a degree of aqueous medium because we're inside the body, so we're talking here about that polymer is going to sort of wrap around to form this sort of helix. So essentially almost like a spring-like wrapping, which then forms a tube. And in that tube, you're going to have this sort of like water channel, right, inside the tube, and then a hydrophobic area, if you like, outside of that. And therefore, that's what we mean by forming these channels, yes?
Yeah, and the channels are connected. They're multiply connected. So they're not just a single channel. They're all connected. Yep. And the degree of connection depends on external variables. They're dynamic and they can change from one form to another. Yeah, that picture's nice. So that's a real analogue formed from what's called a microemulsion, which is it's an analogue. It's got something called a surfactant molecule at the surface between oil and water, which is like the sulfate molecule between the hydrophobic polymer in heparin sulfate and water. So that's we think a real analogue of what's going on. And the same thing happens and you can see the multiple connections there roughly. So you've got water or electrolyte passing through And you've got and for our case, we've got sulfate molecules at the surface of the water between the yellow and the blue regions. And the yellow regions is just hydrocarbon regions of the polymer. So that multiply connected bunch of tubes is the glycocalyx. It's our representation of the glycocalyx. And that passes electrolyte salt water from the blood. The other components of the blood, macromolecules, proteins and so on, are excluded by the exclusion zone.
They're way too large to be fitting in.
And the glycocalyx has, the endothelial surface layer has the property that expels these things.
So I'll get into that in a moment because there's some good examples and particularly some really good videos to sort of show that. But just to clarify, so again, this sort of analog we're looking at here is this sort of 3-part micro emulsion. So this surfactant, in this case, we're talking about, and by surfactant we just simply mean a molecule that has a hydrophobic and hydrophilic ends, if you like. So again, similar to your...
It's like a lipid molecule or a soap molecule.
Yep, like I say. So because of that, it's again self-assembling. It's arranging itself in these structures that allows water to form a channel through the oil, if you like, in this particular case. And so that's, again, because when we look at the glycocalyx cell area, we see the same sorts of molecules that would therefore experience similar interactive forces, and that leads you to believe that not only are these channels forming, but they're also potentially quite dynamic, because we're not talking about PVC pipes coming and locking in together and sort of being rigid. They're a self-assembled structure, so they can be subjected to a degree of dynamic rearrangement. Would that be fair to say?
Yeah, they can expand and extend. They've got less flexibility than a microemulsion, which has got independent surfactant molecules at the surface. For this case, the polymers wind around and they're connected. But so they've got sulfate molecules winding all the way around. But the forces between them, remember they've got sodium coming through and chloride from the blood. The sodium's positively charged, interacts with the sulfate. So they can expand, get bigger, or they can contract and make more connections, depending on the reagent that's coming through. Insulin can come through, get to the surface and expand it, and then let glucose come through. So they've got that flexibility to allow selectivity, to allow some things to get through and other things not. They can let some nanobubbles through, others not.
So again, I'm picturing this sort of spring, right, and this coil, but again, the coil can expand certainly radially, right? You can Python-like digest a large thing, but there's packing constraints. Essentially, eventually that's going to either rip apart or not going to be able to get down that tube. And because of that potential continual expansion and even, I guess, elongation potential of those tubes, but they're all meshed together or packed together a bit more mesh fence-like, if you like, as opposed to, solid wall, for want of a better term. So you've got this network of all these tubes that allow certain things. It's really a traffic control sort of system, isn't it? To say, okay, well, ions can go, well, positively charged ions this way, things that are hydrophobic versus hydrophilic. And I guess then these tubes could be conceptually expanded from the inside, and we'll talk about some examples. You alluded to insulin as an example, but also even compressed from the outside, I suppose, as well, as if the hydrocarbon, the hydrophobic area, if you like, of the glycocalyx gets expanded, or there's different polymers essentially to add additional weight to those areas. At least locally, you can get a degree of, I guess, squashing of those tubes potentially as well. Would that be fair to say? Yeah,
and that's important because it means that the rate of flow through the glycocalyx of salt, and which is important because the sodium and potassium have to be in equilibrium. The potassium's on the other side of the cell membrane.
Inside the cell.
Inside the cell, it's all potassium. outside it's all sodium.
Yep.
So you've got to, you can affect the rate of flow of the sodium chloride solution, outside solution. And that's important for conduction of the nervous impulse with an axon, because the rate of flow of sodium, which determines the rate of signalling.
Yes, okay, right. So again, so what we're talking about here is sodium ions flowing through these water channels, because sodium is going to, be well accommodated inside these channels. But the size, I guess, of that and what else is flowing through that's going to affect, I mean, it's not viscosity per se, but you can kind of think of it in that sort of sense, right? If we have a narrow tube versus a wide tube, if we have a mess of things in there versus a relatively clean, if for want of a better term, a solution, then that's going to drastically, and I guess also the interaction of these of how many substations there are for what, we think of this as sort of almost like a subway network. The more sort of stations there are, then the longer it's going to take things to get through than it is if there's one, a single, express lane, if you like, from one point.
Yeah, and it's actually more complicated than that, because there's about 4 different kinds of these sulfated polymers of different chain length. So you can get shorter chain lengths and...
I appreciate that, but let's stick with it.
I mean, that allows you to appreciate that it's got the flexibility to...
Yes, yeah. And so it could change its function very dynamically, very subtly, very easily as a result of all these moving potential.
Yeah, and indeed that's very important, because the inside of the glycocalyx, it's conducting this salt solution and glucose and insulin and so on, that the tubes are aqueous.
Yes. Yeah.
Outside is hydrocarbon. But for the endothelial surface layer, the long bit that's going from the top, that's teased out, not only is it teased out in these long, thin seaweed-like things there, but those helices go the opposite way. Yep. They're in contact with water, with the blood, and there the helix winds up as a salt with all the sulfate on the outside. The sulfate likes to be in water. So it's very subtle. It can wind this way or it can wind this way. And so that...
And that's an important part, because the reversing of the chirality, again, opens up a whole another level of dynamic channeling, for want of a better term. Yeah. So let's sort of deal with that now, because again, that's kind of what we're alluding to in this picture, right? That some of these strands make their way out into the ESL area, what we're calling the ESL, right? And we'll get into the bubbles, etc., there as well in a minute. But so yes, so within the glycocalyx itself, which is what we're sort of alluding to here, where these sort of, we're getting this helix sort of effect here, creating these kind of water channels. But as these polymers sort of poke out, for want of a better term, up into the ESL, then in this case you're getting a reversing of that. So you're getting these sulfonated head groups, on the outside of these, and so forming different channels and then different variability of the channels from the blood. So up here we're talking about the blood flow, in this case in a vessel, whether artery or vein. And so you've got this, because you've got this sort of systolic pressure, sort of, blood sort of pulsing through, you've got this continual sort of flowing along of there, and then these channels that can effectively distribute, calcium ions, sodium ions, insulin, etc., down through the
can allow it to pass through, the picture seems so incredulous that you're inclined not to believe it, but just recently there's been some good experiments that show you can have this polymer self-assembly with artificial systems that have exactly that property, that one part has got the aqueous core and the other's got the wound up so the water's outside instead of inside.
Right.
So it's like a microemulsion where you have water in oil, oil in water. The polymers can do it too, and they do it. And the analogue with that gives us even more confidence is something called naphthalene.
Yeah. So we'll do a big deep dive into the naphthalene as well, because that's a much larger in terms of scale, but very similar, and that's given us a lot of insight. So I think I just want to touch on, again, this is some of the work from Hans Vink and his...
Yeah, he did a lot of pioneering work over 25 years, and his institute has done a remarkable job in terms of exploiting the properties of the endothelial surface layer to...
So this particular video shows a very, very small capillary. And what you can see from this particular video is that we're seeing, in this case, red blood cells pulsing through that capillary. And you can see here very clearly that there's this sort of gap between the sort of edge of the capillary and where the red blood cells are sort of traveling through. And to give you an idea of sort of scale, and again, he explains this in the video, is that this capillary is actually smaller than a red blood cell. So these red blood cells, this is about a 5 nanometer, I think, and the red blood cells are more like sort of 7 and 8. So 5 microns, sorry, and 7 to 8 microns for the red blood cells. And so these red blood cells have actually been sort of squashed a bit, compressed, so that they can fire along this this capillary. But you can see that there's very clearly almost like I know I often use the analogy of like a maglev train, that magnetic levitation, in that there's clearly some forces here that are preventing those red blood cells from getting anywhere near touching the cell membrane wall, essentially, despite the fact that they're obviously going through here at relatively, the shear forces involved in this would be fairly extreme if you think about, particularly arteries, sort of, pumping that sort of blood around. So,
no, not necessarily, Matt, because the endothelial surface layer it's very sparse, but it's very flexible. It can flow very easily. You can imagine seaweed with the tide going through. Yes. It can lay down easily.
Yes.
But if you try and squash it, it's going to oppose it. Yes. Because these things in the stationary case have got very large long-range forces between them that people haven't recognised before. And those large forces are cooperative, and they stop anything like a blood cell or other from going. So they've got the dual, very remarkable property. They can flow very easily and lay down, or they stand up, and I'll get into you if you...
Yeah, so I sort of picture that as, again, and if you think about it, as a way of dealing with the shear forces that would be there if that layer wasn't there. Yeah. So, in fact, I find it very similar to the way geckos, if you look at the way geckos are able to attach to surfaces, they have these kind of, look to the minute detail of what the geckos have on the ends of their hands or feet, whatever they are. They have these sort of series of little hooks that kind of lie down. And then when they go to move, the surface area is now so extreme that it sort of grips, and they're able to use those forces to literally climb up walls, because they have this sort of like peeling on, peeling off scenario that's energetically really, really...
Yeah, it's the same, but they're different forces in that case.
Sure, but I guess in my relatively simplistic mind, it's, I guess what I'm alluding to is nature's found a way to energetically and very carefully disperse those forces, or not suffer the consequences of, say, how do we fire this thing down a tube, at huge speed without massively, disrupting the tube itself, and particularly obviously this very tiny sort of cell membrane. So I guess again, what I wanted to sort of hone in on here is this concept of the ESL has a number of functions, and we'll explore in detail. But one of the very clear ones is this idea of this exclusion zone, creating this barrier to ensure that, yes, there's a whole bunch of tubes and this sort of connected network to allow mediation towards the actual cell membrane itself, but very clearly exclude, red blood cells and white blood cells and anything else that's in the serum itself that we don't want realistically anywhere near there. It's very much a sort of a separation sort of function. Okay, so that's they're sort of, I guess, some of the key structure in terms of the components of it. So again, just to sort of recap, what we're suggesting here is that the ESL GC really is based on self-assembly processes, but it's a dynamic, if you like, biological structure that connects virtually every cell in the body, particularly the cells that are, I guess, permanently connected, as well as cells, like red blood cells, etc., that are, or bacteria cells that are sort of, floating through on their own, if you like. That nearly all of these cells have this structure, and it's there to form to function a number of different very, very important roles. And in fact, what we'll allude to as we go into this a bit further is this idea that it's the glycocalyx itself that ultimately underpins what we might even refer to as homeostasis. Is that if we don't have a properly functioning GC-ESL system, then many of the things that we would ultimately refer to as diseases or interruptions.
If you've got a damaged glycocalyx, then a cancer cell can come up to it, or a T cell, and it can penetrate into the tissue. But if you've got a healthy glycocalyx, you can't have a low-density lipoprotein forming on top of the cell surface or membrane surface and messing up the flow of liquid, and again, you lose the protection. So any kind of deposition is going to mean that you're sick.
Yeah. And again, when we go into a bit more detail, particularly when we look at things like atherosclerosis and some of those other issues that we'll deal with as we go through, again, Vink's shown he's got some very, very good videos and very good work to show, in almost sort of acute collapsing of the ESL, you can see the effect of, white blood cells moving into the immune response, right? And literally, rather than sort of rolling along their merry way as they normally do, that they're going to find these holes in the ESL, if you like, and then start to generate a immune response and inflammation, and even sort of cholesterol leaking of LDLs, etc. So we'll certainly look at that in a lot more detail. But I think from this sort of, I guess, initial proposition is that what we are talking about here is a biological structure that has a degree of complexity in it, but it is based on some very, I don't want to say simplistic, but some very well understood self-assembly principles. And we've got some good analogies. We talked about NAFI and some other things we'll look at when we look at, well, what makes you so sure that this type of structure is in place? We'll look at those, and then we'll also look at a whole bunch of implications that might have. We'll be looking at things like how insulin sensitivity and adipose tissue forms as a result of looking at these structures, how things like acupuncture can be explained. We also looked a little bit in terms of lithium and the effects on things like bipolar disorder and propagation of signal, and even, to how certain drugs work and the interaction of drug molecules with.
Well, nearly all drugs are involved in changing the glycocalyx.
And changing the structure and therefore its function, yeah. So that we'll deal with that next. Is we'll start to look at some of the implications for that structure. But first of all, we'll go into a bit more detail, I think, around how the evidence for the analogies, and particularly we'll do a bit of a deep dive into NAFI in particular, because that's a very good example, a very more observable example, if you like, of why you believe that the structure of the glycocalyx ESL is as it is, and what implications that has for this. So we'll get into that next.
Okay.